Fine.
Ini teh berapa kali, Ran?
Sekali.
Oh, sekali ya.
Atau salah? Cekap.
Atau sekali. Kami wae sekali sareng 10 kali.
Mohon.
Aduh.
Yeah, we're just coming out of lockdown here, Rani. So kids are still homeschooling at the moment, not completely back to school.
Yeah.
Okay.
I heard from the news that it's like kind of free day now in Melbourne, right? We can have coffee in the cafe, but school still online.
Some days are, but as of next week, it'll be back full time for them.
Oh.
So it's like half a week. For the last 2 weeks, they've been going in and out of school, and today's unfortunately not one of those days. But we're almost there.
Yeah.
But myself, I've still been working from home. I haven't been in. I think I've been on site probably 6 days in the last 18 months.
Yeah.
So it's been difficult for everyone, I think.
Yeah. I think it's almost 1 month in Indonesia that school has been open, but with some arrangement. For example, for this week they will go to school, but next week they will study at home, or any other arrangement. I mean, it's not really full, but still some limitation.
Yeah. I'm sure it'll be like that for a while.
Yeah.
Till next year, I suspect.
I think so. Yeah, until next year. Yeah. Okay. So we are still waiting for another 1 minute, yeah, Brad?
Yeah, that's fine, Rani. So is this a forum that students can intervene and ask questions, or do they ask questions at the end, or how does it work at the end? Okay.
Yeah, I think you can complete your lecture first, and then there will be a session for discussion, so student can ask question. Is that okay? Yeah.
That's fine. I'm fine with that arrangement. Yep.
Yeah. So I think it's already 1 p.m. Yeah. Can we start, Brad? Yeah. I think other college or other lecturer that will join us will come pretty soon. Okay. Bu Ika, kita mulai aja ya, Bu Ika.
Yeah, okay.
It's already 1 p.m.
Alright.
Yeah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Yeah, good afternoon to Dr. Brad Sleep in Melbourne, and good afternoon to all of the participants, students, lecturer, and other college that have joined us today in guest lecture of selected topic in organic synthesis. So today's lecture is quite special because we have a guest lecturer from Walter and Eliza Hall Institute as our international collaborator that will give us a topic, an interesting topic, which is drug discovery in infectious diseases. So before we are listening to Dr. Brad Sleep's lecture, I'm going to read the brief CV of Dr. Brad Sleeps. So Dr. Brad Sleeps lives in Victoria, Australia. He did his bachelor degree at La Trobe University, majoring in chemistry and biochemistry. So after completing his bachelor degree, he took honors and PhD program at the same institution at La Trobe University, Bandura. under the supervision of Associate Professor Andrew Hughes. This is also my supervisor. So after obtaining the PhD degree, Brad has done several casual and part-time before he joined WIHI in 2005 as research officer. So from 2007 until 2018, Brad has become a senior research officer at WIHI, and from 2018 until present, he's currently a laboratory head of chemical biology division at WIHI. There are so many publications that have been resulted from his research. His research has been published in many high impact factor journals, such as Journal of Organic Chemistry, Journal of Medicinal Chemistry, Bioorganic Medicinal Chemistry, and I think there's some more journals such as PLOS ONE, Molecules, and even there are about 15 patents have been resulted from his research. So besides his research experience, Brad also has many educational experience, such as he has been a session lecturer at Department of Chemistry at Trump University from 2009 and 2012. And also in 2019, he has obtained Max O'Connorship lectureship from La Trobe University. So if you want to see any papers that have been published by Dr. Brad Sleep, you can also search from online references such as Google Scholar, Web of Science, and many more. So I think that's the brief CV of Dr. Brad Sleep. We are looking forward to hearing his lecture, Drug Discovery in Infectious Diseases. To Dr. Brad Sleep, time is yours.
Great. Thank you, Rani, for the nice introduction. I'm very glad to give this lecture on behalf of my laboratory, of which most of the work that you'll see today has originated from. And I believe you've... Thanks, Rani. And I believe you've already met at least 3 of the lab members who gave lectures earlier in your curriculum, Trent Ashton and Curry and Lydia Scott. So I hope you enjoyed their lectures. So I'm not sure if they gave you a little bit of a background behind our institute, but I thought I might give a touch of the background behind our institute and what we do here. And then I'll move into a bit of a background behind that drug discovery before moving into examples of, I suppose, small molecule development in my laboratory in infectious diseases work, which obviously segues into synthesis of bioactive compounds, which of course is the key topic of this seminar. So at our institute, just to give you an idea of where we're located, as Rani mentioned, so we're in the southern part of Australia, in a city called Melbourne. I suppose comparably to cities in Indonesia, we're relatively small by population. So we have approximately 7 million people in Melbourne, but compared to cities in Indonesia, the city's very spread out. It covers a lot of space and volume. And so we have 2 main campuses at WIHI. The first is situated near the central business district, as you can see here in the background, which overlooks what's known as Port Phillip Bay. And approximately 20 kilometres north, we also have what's known as a biotechnology site, which houses aspects of our institute's research, such as antibody research and also medicinal chemistry. So at our institute, we have several key disease foci. starting off with infectious disease. So we have a very strong focus on malaria, but we do also delve into tuberculosis and HIV and some other parasitic infections, as I'll touch on. We also have a very strong immunology presence, so much so that two professors from our institute have won Nobel Prizes for their research in immunology in the past. And so with that strong presence in immunology, we focus on autoimmune diseases. We also now have a very strong oncology presence in immunology also. We also focus on inflammation, particularly diabetes, rheumatoid arthritis, and celiac disease. And of course, one of the Institute's very strong capabilities is in cancer research, particularly blood cancers, such as leukemia, also lymphoma, breast cancer. And we're starting to reach out into colon cancer and very recently brain cancer. And this is really this research is really strongly underpinned by expertise in structural biology, chemical biology, or medicinal chemistry, which is also houses organic chemistry. We also have a very strong high throughput screening facility, which I'll come to in a moment, as well as bioinformatics, such as genomics and proteomics, for example. So I thought I'd start today's seminar by moving into the drug discovery process, particularly looking into the starting point identification. And so here today, I'll only be talking about small molecule drug discovery. I won't be talking about biological drug discovery. Obviously, small molecules fairly relevant to today's topic. And then I'll move into how we go about to optimize these molecules in terms of the structure-activity relationship, but also touch on physiochemical properties and ADME, which is in some ways lost at a very basic drug discovery fundamental point of view. And then I'll go into certain case studies from my laboratory in the synthesis of those bioactive compounds in malaria, schistosomiasis, and HIV. So first of all, starting off with the drug discovery process. Here we have a schematic showing the drug discovery process, where we have several key points along that process on the way to generating a drug that makes it to the marketplace, so suitable for use in humans or widely in the population. And all drug discovery really starts and is underpinned by basic fundamental scientific research that identifies either the protein target for the drug discovery process to target, or some type of disease phenotype for the drug discovery process to target. Then we we move into the drug discovery phase, which includes hit discovery. And hit discovery is all about trying to identify a small molecule starting point for medicinal chemistry to then go on to optimize. And generally, this comes from target and phenotypic-based screening approaches, which I'll come to in a moment's time. And then we move into lead generation, which is generally optimization of these compounds based on in vitro potency, but also off-target selectivity, looking to move towards activity in some type of in vivo model. And then lead optimization is taking a lead compound or lead compounds from the first phase. And then we look to improve the in vivo efficacy of these molecules in lead optimization phase, but particularly concentrating on the safety aspect, because we really want to have a compound that moves into human clinical trials that is very safe and robust. Preclinical assessment phase, which is where we have the drug assessment phase, is basically submitting a dossier to a regulatory body so it can get approval to move into human clinical trials. And so in human clinical trials, the first phase is all about determining the pharmacokinetic profile in humans and working out a dose administration route and also dose, which should have been predicted in lead optimization phase, but also looking for efficacy in some type of model usually in healthy individuals. And then phase 2 clinical trials is all about testing that molecule in humans that actually have the disease. And phase 3 is in a very wide cohort of individuals across a diversity of backgrounds. So that's the drug discovery process en route to taking a drug to the market in a very brief sense. But I thought I'd start off by just briefly talking about starting point identification, because that's going to be a key to some of the case studies that I talk about later. So in identifying small molecule starting points, at our institute, at least, we generally take on what is known as high throughput screening of a large chemical library. And you can do this in 2 ways. You can do this in a cell-based or a parasite-based, in this instance, phenotypic approach, or you can do it against a protein target. So first of all, I'll talk about the small molecule screen against a cell or a parasite. So generally, we want to identify some type of high throughput screening assay that will be able to detect cell killing or some type of cell phenotypic readout that is required to identify that small molecule. And then we screen that small molecule against a large compound library, and those libraries I'll touch on in a moment's time. And once we've identified that small molecule, we then move into optimization of that hit class small molecule against the whole cell or parasite readout or assay. And this process is known as a rational design, where we don't know the actual how the compound binds to the molecular target. And so that's one of the disadvantages of screening against a whole cell or parasite, that we don't know the actual protein target of what we're designing a small molecule against. So in parallel, what we usually do is try to identify this protein target of the small molecule class using various methods, such as resistance studies or some type of chemical biology approach. And those methods I don't have time to go into today. But once we've identified that protein target, we usually express the protein target, which then could enable a structure-guided design approach, in that we can get an X-ray structure of that small molecule bound to the protein target, which expedites small molecule optimization. And this feeds back into our optimization of a small molecule on way to developing a drug candidate. So that's the phenotypic whole cell approach. But the protein target approach obviously has its advantages in that the screen is done against the protein target. But to get to that protein target before we commence the high throughput screening, there's a lot of background work that's done at a fundamental level to understand the role of that protein target and whether it's essential to the disease that we're targeting. So in doing so, we must validate that protein target using a number of different genetic models, usually knockouts and knockdowns, etc. Then after that, you must be able to express that protein target, develop an assay that is amenable for high throughput screening against that protein target before you actually commence the screen against the protein target. So there's a lot more work earlier on to do a high throughput screen against the protein target. And then, of course, once you've identified the small molecule that binds to the protein target, is that a problem, Rani, that the recording stopped?
Yeah, I think the host is left, but I can record it again. Yeah, is it okay? You can continue your lecture.
No problem, Rani. Thank you. So then we go on to optimize the small molecule hit class against the protein target This should say here, that's my mistake. This is what is known as a structural guided design, because we may have the X-ray structure of the small molecule bound to the protein. And then, of course, at some point in time, we want to evaluate that small molecule that's been developed against the cell or the parasite of interest, which is related to the disease, before going on to the drug target. So in terms of performing these high throughput screens, we'd usually screen against a small molecule library. So these small molecule libraries contain small molecules that generally have a molecular weight of greater than 200 and less than 500 in molecular weight. And these libraries are usually derived from a chemical vendor. And there's many chemical vendors available that specialize in providing compounds for high throughput screening libraries. And so these libraries, generally an academic institute like ours, range from 50,000 to 250,000, or in that order. But if you go to a pharmaceutical company, it's not too out of order to have a screening library that's in the order of 2 to 4 million compounds, and they can do that because they've got a lot of resources that an academic institute certainly doesn't. And so the hits that what we call hits, the compounds that are identified from the screen that are active, are generally in the range of 500 nanomolar, but certainly less than 10 micromolar in range. And as I mentioned, the assays here can be either a biochemical assay for a protein target or a cellular type biomarker for a phenotypic assay. There are other screens that you can do that have really come to the fore in the last 20 years, but these are only against the protein target, and they're known as a fragment-based screen. So typically these fragments have a molecular weight that is much smaller than the small molecule library, between 100 and 200, so really quite small. And they sample quite a large chemical space in terms of diversity, because they're smaller in size. And that's typically why these fragment libraries are between 1,000 and 4,000 fragments in size. And so the hits that come from these fragment-based screens are typically in the order of 250 micromolar to 1 millimolar in range, so significantly higher than what you'll get from a screening library. And the reason for that is because they're quite small, and that's the reason why their affinity is so much lower than it is for a high throughput screening hit. Primary screening assays, however, must be adjusted, because typically up at this activity range, your biochemical, it's out of the range of a biochemical assay. So typically the primary screening assays for a fragment-based screen are either NMR, SBR, probably not ITC, but thermal shift, I think, will come online soon. Very much so mass spectrometry in the last 5 years. And you can also do this by direct X-ray crystallography. And the final point that I like to make is to identify small molecules against a protein of target of interest. You can actually design mimetics of the natural substrates that bind to that target of interest. And I'll give examples of that a little later on. So that's the starting point identification, very briefly. And so now I'll move into the optimization phase. So once we've identified that small molecule starting point from the screening process, we begin what's known as a structure activity optimization, where we start to improve the potency of the molecule. But we first must understand how or what components of the small molecule are required or obsolete for activity. And this is where we move into the lead generation and optimization phase. But you really want to fundamentally understand the structure activity relationship at a very early hit to lead, hit discovery phase. So to optimize these molecules, is a multifaceted approach, and it really requires a large team to do this correctly. And so here we have a very, I suppose, vague look at a drug discovery evaluation cascade, starting off with the primary assay, which could be either biochemical or a cellular type assay. And once we've determined the activity in these assays, we usually have other assays to support it, such as an off-target cytotoxicity assay or an off-target protein assay. We also have, if we know the structure, structural biology informing medicinal chemistry on how to improve the molecules. We also have other secondary, either protein assays or generally cellular assays that also inform medicinal chemistry. We also have different ADME and toxicity type assays to inform medicinal chemistry about moving into an in vivo model, such as a mouse model shown next. And if we see efficacy in these models, then we'll move to other types of secondary animal models and toxicity models before going on to a lead candidate. And this process takes at least 24 months, and it's not uncommon to have at least 6 medicinal chemists on the project like this at an early stage. And once you move into lead candidate phase, it's not in a pharmaceutical company, it's not uncommon to have anywhere between 10 and 30 chemists working on a project at once, really driving it forward as quickly as possible. But just to do this whole drug discovery cascade takes a lot of different personnel and with different expertise, as shown here. And so you can imagine how resource intensive this process becomes. And that's certainly why at our institute, in a very early stage, we try to collaborate with a pharmaceutical company as early as possible, because we certainly at our institute don't have all this expertise, nor do we have the resources to develop a drug candidate. And so collaborating with a pharmaceutical company or a biotech company is utterly required. And so compound optimization is an iterative process, as mentioned. So obviously medicinal chemistry is key to driving the optimization of these molecules, but we seek feedback from testing cascade that I just showed on the previous slide, which obviously entails either biochemical or cell-based assays, ADME-based assays, which I'll touch on a little later on, and in vivo models, before getting that data analysis and that feeding back to medicinal chemistry, as well as any structural information that you may have of your compound binding to the protein target of interest. So I thought I'd just touch on structure-activity relationship. And so this is what defines what motifs of a small molecule structure are required for or are deleterious to biological activity. And it really allows the medicinal chemist to tune in and drive biological potency of these small molecules. And so the structure-activity relationship, or SAR for short, is the relationship of molecular functionality or groups on that small molecule and its relationship to the biological activity. And so key drivers of that structural activity relationship are hydrophobicity, steric volume, electronic effects, hydrogen bond donor acceptor interactions, but also rotation around certain bonds with different attached motifs. And these SAR principles can be applied to both target-based and phenotypic-based approaches. And so I thought I'd give you a very simple example of SAR. And this research was undertaken by mostly by undergraduate students, to give you an example. And so generally when we get hits from a high throughput screen, we like to have nice simple hits to work on, and that allows the chemistry to be performed very quickly. And And so most of my talk, as I mentioned before, is going to be on infectious diseases, in particular targeting the malaria parasite, which I'm sure you're all familiar with in Indonesia, but I'll touch on that in a moment's time. But for this talk, this is the, for this aspect rather, this is the high throughput screening hit that we got from a high throughput screen against this malaria parasite. So this is the top screening hit. So 250 nanomolar potency, which is fairly decent. So what we wanted to do is, in this structure-activity relationship, is determine which motifs are most important for activity, or even deleterious to activity. So first of all, starting on this 4-amino substituent of this quinazoline scaffold. And you can see here quite quickly, if we change to a dye substitution here, we almost ablate or significantly lose activity, which suggests to us that dye substitution's not tolerated. So that NH must be making some important interaction with the protein target within the malaria parasite. You can see here, if we change this mono substituent to this tethered furan or to a methyl group, we don't really see much change in activity. So that's suggesting to us that substituent's not all that important for activity. But perhaps we could utilize that area in future for modifying physiochemical properties. Then we look at the 2 substituent, and again, you can see dye substitution certainly impacts activity. So obviously we don't want to do that. It seems that aliphatic substituents don't affect activity. And again, I'm just showing a small subset of examples here. And it seems to be that a CH2 and a phenyl ring are accepted. But what we found more often than not was that this direct attachment to this arrow ring was optimal. And so looking around substitution around that arrow ring, we see that the meta substitution seems to be okay in terms of activity, but when we move to the ortho substitution, we lose activity. And again, 3,4 substitution given seems to be okay. So that suggests that the para substituent seems to be okay for activity, at least with these methoxy substituents. But if we substitute in some more polar substituents, again, we typically lose activity somewhat. But if we substitute in some electron withdrawing groups that are quite small in nature, such as these halogens in the 4 and the 3 position, we can see we start to drive potency towards 100 nanomolar potency, while the ortho substituent, we lose a little bit of activity. And so we further looked at dye substitution on this arrow ring, and again, we were able to drive this down below 100 nanomolar potency now. Some iterations, such as endocyclic nitrogen in the ring, seem to be tolerated in some instances, but not in others. So we just maintained the simple arrow ring with subsequent analog sets, and then we looked at the back ring system and substituted another heterocycle, as can be seen here. And mostly these were detrimental to activity, apart from this one endocyclic nitrogen in this position. But nevertheless, we maintained just with the basic arrow ring with other substitutions. And what we were looking to do here was install some polarity into the molecule to increase the aqueous solubility. And so that's the reason why we installed in these other polar aliphatic groups into the 4 position. You can see now we're driving activity down to the 50 nanomolar range with some of these substitutions. And just to note that we didn't have any very little cytotoxicity against the human cell line, which means we had a fair window of selectivity. So we took the best substituents in the 2 and the 4 position, combined them into this focal set of molecules here, and now we're down to 20 to 30 nanomolar potency, which is similar to that of the known drug chloroquine. And so this is just a very simple example of how structure-activity relation optimization plays out. So I thought I'd just touch on physiochemical properties, as I've mentioned very briefly in that SAR example, but also work into ADME a little bit as well. Some of these factors are forgotten in simple medicinal chemistry design. And so designing or optimizing a small molecule is not all about just driving biochemical or cellular potency. There's many other facets involved. So just as an example, if we're looking at in vitro biochemical potency, you have to worry about selectivity against other protein targets, but also the aqueous solubility of the molecule in that assay. It's no good having a molecule that's not soluble in aqueous media, because that might give you a false readout in your assay. But when you move into a cellular context or perhaps a parasitic context, as it is in my research, you also have to then worry about cell toxicity, so off-target effects in the cell. You also have to worry about whether your compound is cell membrane permeable. You have to also worry about with your protein, so your compound's being turned over metabolically in that cell type. You also have to worry about non-specific protein binding, particularly given that the media your cells are in contains a lot of albumin. Then when you move into animal studies, you have to also worry about additional factors such as stability in the gut, stability in the blood plasma, whether or not your compound's intestinally absorbed, how systemically exposed your compound is, what your compound is doing to the body in terms of pharmacokodynamics, which I'll come to in a moment's time. And then you have to worry about off-target polypharmacology, plus also toxicity that you might observe. So it's just not all about driving potency at a biochemical level in optimization. There's a lot of other factors medicinal chemistry has to consider. So some of the physiochemical properties that you must consider are quite simply, one is polarity, which is related to the quantity of polar atoms in your molecules, such as nitrogen and oxygen. And a very simple in silico calculation that anyone can do is the polar surface area, which is a calculation of the polarity installed in your molecule. And this is important, as I'll touch on in a moment's time. So lipophilicity is the other, which is effectively the converse of polarity. And so this is related to the quantity of hydrophobic atoms, such as carbon, sulfur, halogens in your molecule. And this can be very simply measured by what is known as the partition coefficient, or log P. And so this calculation is generally not pH dependent, but it's a very simple in silico calculation that can be done by many online programs. And you can measure this experimentally also, but not just in silico. And usually you do this by partitioning your compound in octanol water and then determining the log P by HPLC. So a very simple example, and certainly not a drug by any stretch of the imagination, on the interplay between polarity and lipophilicity. Is given here. So we see that a molecule with no with no polar atoms in it generally has a very low PTSA, whereas the C log P is generally high. But as soon as you start to substitute in these polar molecules, the C log P goes down, the PSA starts to go up with these calculations. And certainly if you think about pH dependency, some of these molecules, these nitrogens may be protonated at a certain pH, which certainly affects their lipophilicity readout. And so this is where log D calculation comes into play. And certainly it's important to consider when the molecule contains either acidic or basic functionality. And so why is polarity and hydrophobicity important in a drug? So really the balance between polarity and hydrophobicity determines its aqueous solubility, whether it's membrane permeable, whether it's going to be highly protein bound, whether it's going to have these promiscuity effects such as undesirable off-target interactions. And these are all important factors in generating a cell-active and in vivo active compound. And so these, what is known as drug-like properties, was loosely defined many years ago by Lipinski's rule of 5, but there's now many, many other metrics that have been introduced since then. And so Lipinski's rule of 5 states somewhat that a small molecule must have a molecular weight below 500, a log P between 2 and 4, and a PSA of between 80 and 120, plus a couple of other factors. And so this rule was introduced to give medicinal chemists an idea of the parameters your molecule must have, ideally to become an orally bioavailable molecule. But of course, there's exceptions to this rule, but it's a good rule of thumb to play by when designing a small molecule. And so those are the physiochemical properties. I thought I'd just touch on ADME as well. So this is when we move into an in vivo system. So beyond compound design and synthesis, when you go to test your molecule in an in vivo context, you want to know what your body is doing to that compound of interest. And this is known as absorption, distribution, metabolism, and elimination, or largely known as pharmacokinetics. So this is effectively how this compound is absorbed, distributed, metabolized, and excreted from the in vivo system once administered to it. But there's also another factor, and this is what the compound is doing to the body in terms of its efficacious, whether it's selective, and also its potency. And this also plays on with selectivity and, toxicity here also. And this is known as Pharmacokodynamics, or PD. So just having a look at pharmacokinetics, because I thought this was important. So when you administer a drug, you want to know the barriers to your drug's action. And so you can administer your drug to your body in many different ways. So of course, there's intravenous, which is straight into systemic exposure into a blood vessel, but there's also oral administration. But you can obviously have intranasal, you can have eye drops, you can have ear drops. You can have many ways to administer your drug to the body, or at least the tissue of which the disease is most prominent in. So just to give you an idea of the barriers to drug action, if you administer your drug intravenously, obviously it goes straight into systemic circulation, circulated by the heart, but it will sometimes go to the target tissue, and other instances it will bypass the liver, which is obviously the metabolic powerhouse of eukaryotic or mammalian systems, should I say, that turn over the compound of interest relatively quickly. But if you're going by an oral route of administration, you have an extra barrier of for your drug to make it into systemic circulation, which is the gastrointestinal tract, before going through the liver, again, being turned over, and before making its way to the target tissue. And if you're working on a central nervous system target, you obviously have a blood-brain barrier issue to overcome as well. So just to give you an idea of what pharmaconomics is, it's really to look at the systemic exposure of your compound when administered to an in vivo system. So here on the y-axis, we have the drug concentration in systemic circulation versus time. And so if you administer your drug intravenously, it goes straight into systemic circulation. So you get a very high initial burst of your drug systemically exposed, which is usually read out by Tmax somewhat, and then it's slowly metabolized and it's secreted over time. But if you administer your drug orally, it's obviously got a slow intake in terms of systemic exposure, because it has to make its way into the intestinal tract, bypass the liver before it makes into this systemic exposure. And the point of which it has the highest exposure is known as Cmax. and also the time to take to get there is known as Tmax. And so there's other important factors here in measure, and obviously we're just concentrated on absorption here, but there's distribution, so that is effectively area under the curve here, which is related to metabolism and elimination over time. And so it's very important knowing how much of your drug is systemically exposed, because it's no good having a drug that's not systemically exposed that is well below the in vitro EC50 or ED50 value. So you want to have your drug systemically exposed significantly above, usually 10 to 100-fold above the EC50 value of the compound being developed. And that's the reason why pharmacokinetics is so important. It's also important to know how long your drug's on board before giving perhaps another dose of your molecule. So there's other factors, as I mentioned, which don't have time to go into here, but I thought I'd just mention them very briefly for you to go away and have a look at if you're interested. And so there's a determination of in vitro absorption, and there's a number of different models that can determine that. And it's very important to know how your physiochemical properties relate to in vitro absorption, as indicated on this slide here. There's also metabolism, of which you can determine in an in vitro context before going into an in vivo system. And this just explains the metabolic process that happens in the liver for you to understand that process. And here's how you determine this metabolism in an in vitro context and the method involved in that. And finally, distribution and protein plasma binding, which is important in knowing how your compound partitions in systemic exposure as well. And again, there's different references here for you to read up on if you're interested in knowing more about that, but all important factors in designing a drug, as well as toxicity and adverse reactions, which can be tested in vitro, which assist in de-risking your molecule as it advances further into human studies. So now I'll move on to bioactive case studies and the synthesis of bioactive compounds using key examples from my laboratory's research, starting off with malaria. But before I go into that, sorry, I'll just focus on the overarching goals of my lab's research. And we also focus not only on those infectious diseases, but also on cancer. But two of the goals are listed here, and that's to design small molecule probes to investigate the importance or the essentiality of biological pathways or proteins in disease pathogenesis. And the second is to translate those basic findings towards the clinic. And as I mentioned before, the first one we can do at our institute. The second one, we certainly don't have the resources to perform this task at our institute. So this is where we need to collaborate with a drug discovery, a large pharmaceutical company. And so I'll come to our an overview of our research in a moment's time, but I thought I'd touch on malaria disease very briefly, because that'll be the large focus of a certain subset of talks coming up. So the malaria parasite infects humans. The most prominent forms are P. falciparum and P. vivax. There's a lot of cases globally, and unfortunately this disease still results in many deaths, in particular in sub-Saharan Africa. And the reason why this disease is so difficult to target is because it has a complex multi-stage life cycle involving a mosquito host that can transmit the disease from human host to human host. And I'll touch on this life cycle a little bit more later on. And so the existing treatments are really these drugs that have been around for quite a long time now, such as the quins, or such as chloroquine and primaquine. but also the more newer artemisinin class drugs. But there are now emergence of resistance against all these drug classes, and that's why it's important we develop new chemotypes against the malaria parasite. And so some good progress has been made in the last 15 to 20 years, and some of the key molecules are now going under clinical trials targeting new drug targets within the malaria parasite. And these compounds have different activities against multiple stages of the parasite's activity, which is important in different aspects, such as developing a prophylactic or a transmission blocking drug, as well as a curative therapy. And so we reviewed this a little while ago. And as I mentioned, this is promising as these chemotypes going through phase 2 clinical trials. But moreover, there's still a need to develop new therapeutics with novel mechanisms of actions against these parasite stages. And so just to give you an overview of what my lab is doing at the moment, I suppose this is a snapshot in the infectious disease area. We work with both other academic institutes, but also pharmaceutical companies such as Merck and Janssen towards developing these antimalarial therapies. And I thought I'd touch on several of these today, as indicated here. Several of the other ones I can't touch on because of confidentiality issues. So I thought I'd start on by touching on the ones in collaboration with the Burnett Institute. So we've already introduced this molecule from the screen that we did before with the SAR study. And we were able to get a molecule down to 20 to 30 nanomolar potency against the malaria parasite. It had some activity against the transmission phase of the malaria parasite, but also the delivery phase. We're also able to cure mice with these compounds at a relatively high dose, which is promising. But now to the synthesis, so retrosynthetic analysis of this molecule. You can see I've made 2 disconnection points, which I think in your curriculum you're familiar with, retrosynthetic analysis. So here you can envisage the formation of this carbon-nitrogen bond here by a nucleophilic attack of an activated heterocycle, and likewise down this 2 position. So quite simply, we take commercially available building blocks such as 2,4-dichloroquinazoline and perform a arithmetic nucleophilic substitution reaction, which is simple displacement of that chlorine atom. And this happens relatively selectively in the 4 position, given the tomerization, the stability of that intermediate there, which I'm sure you recognize from your studies. We also have a presence of a base in this reaction to mop up the excess HCl that is created as a byproduct. And this reaction goes quite smoothly at room temperature, so no need for heating here. So it's quite facile, given how activated that nucleophilic center is here. So the second reaction is a little less nucleophilic because of 2 reasons. One, because this substrate is now being deactivated because it's got an amino group here towards nucleophilic attack. But 2, this aniline is not typically nucleophilic. So we need to activate this system in some way. And the way we do that is put in an acidic media, if you like, and we put in here trifluoroacetic acid, but it can use HCl. And quite strangely enough, what this does is protonate this quinazoline ring towards nucleophilic attack. So further activates the system. And we require quite high heating, and we use microwave radiation here because it's a little bit quicker than just using thermal heating. And that's the reason why the reaction only takes in the order of 15 minutes. We get out moderate yields to this molecule. And so that's the synthesis that we use to generate these antimalarial compounds. So another screen we performed in collaboration with Paul Gilson at the Burnett lab was to look at compounds that block invasion of the malaria parasite into the erythrocyte in asexual blood stage. And from that screen, we found a molecule such as this one here, known as this phenylsulfonol piperazine antimalarial. And we're all able to optimize this molecule somewhat to give this compound known as S38. And you can see here that the SAR was actually quite tight, given that you can see the similarities of this part of the molecule with the screening kit. But we're all able to manipulate the compound in this area here to increase activity. And what you may note is that the S enantiomer here is the active enantiomer, whereas the R was less active. And we're also able to perform resistance of mechanism of action studies using drug pressure studies. And this enabled us to generate resistant parasites and then go into genome sequence the resistant clones and revealed a single nucleotide polymorphism in profilin, which is known to interact with actin, which is a key protein which is involved in the motor that drives the myrizoite, the form of the parasite, into the red blood cells. So highly consistent with the mechanism of action. And we're further looking into that. So moving on to the synthesis, we can see we have 3 different condition connection points in this small molecule. You can imagine formation of this sulfonamide via some type of nucleophilic attack onto an electrophile, such as the sulfonol chloride, formation of this carbon-nitrogen bond via simple nitrogen alkylation, and formation of this carbon-nitrogen bond via some type of amide coupling. And so starting with commercially available starting materials, so this sulfonol chloride and this BOC protected piperazine. So BOC stands for tertiary butyl carbonyl protecting group. And so quite simply, it's just a nucleophilic displacement of this chlorine atom in the presence of base to mop up the HCl byproduct generated. And this is relatively facile and done at room temperature effectively. Then we want to remove this BOC-D protecting group. So I think you've been introduced to the tertiary butyl carbonyl protecting group before, and its liability towards strong acids such as trifluoroacetic acid. And this is really driven through an elimination reaction generating isobutane gas, but also CO2 gas is the only byproduct of this reaction to give the amine in this instance. So we have this building block in hand, and we need to build up the other side. So we wanted to generate this chiral center here. So we turned to the chiral pool. So this is just a methyl ester of lactic acid, which is a chiral a natural progenitor. We then activate it with a trifolic anhydride, which is just a simple nucleophilic attack of that oxygen on the anhydride to form this trifolate, which is effectively a surrogate for a halogen, if you like, as a leaving group. And then we perform, and this should be, sorry, my mistake, should be an amine group here. So this building block here performs a nucleophilic attack on this trifolate in an SN2 process. So that's why you get an inversion of this stereocenter to give you this molecule here. And then, of course, we want to remove that methyl ester. And typically we do that under base hydrolysis. But given that we would have had a zwitterine here to isolate in that workup, it would have been tricky. We would have lost some of it in the aqueous media. So we decided to do a acidic hydrolysis of that methyl ester, which allowed us to isolate this quite simply as a HCl salt for the following reaction. And so we then perform what is known as just a simple amide coupling reaction using what is known as HBTU. So this reagent is effectively a hydroxybenzotriazole-like reagent, which forms a hydroxybenzotriazole activated ester in situ. in the presence of dipia, which is a base. And so this amine then acts as a nucleophile onto that activated ester to form, and obviously the activated ester leaves the leaving group to form that carboxamide bond to give you the final product. So then I think we'll move on to some of the work we're doing with Janssen that we've recently published. And this was performed by a PhD student in my lab, Brody Bailey. And so this is the hit compound that we got. So it's got this triazolo pyrimidine type scaffold with this carboxamide and this unusual arrangement on this aryl ring. And so again, it's relatively potent for a hit compound against the asexual stage parasite. She was able to optimize this compound down to 50 nanomolar potency. And furthermore, she again performed a structure-activity relationship, showing the importance of the dimethyl substitution in terms of activity. We also showed this entire moiety here was important for activity, and any change was deleterious to activity. We also showed that this group here was, I suppose, had a number of functional groups which were tolerated, but CF3 was the best. And hydrophobic aliphatic systems here tethered by a nitrogen were most tolerated, such as a piperidine ring. And we're able to show that this molecule interfered with the tropozoite stage of the asexual blood stage and had a very moderate to slow asexual rate killing in terms of its relationship to other antimalarial drugs. So moving on to the retrosynthetic analysis of this molecule. So from commercially available starting materials, you can imagine a disconnection between this carbon and nitrogen bond here, so some type of aromatic nucleophilic attack, and also an amide bond formation here. And also show you the brief construction of this heterocycle. So to generate this molecule here, we start off with this nitroaniline, which has a two substituted halogen. And so you can envisage, given it's got a nitro group and a CF3 group here, this system is relatively withdrawn for nucleophilic attack. And so with gentle heating, you get displacement of this fluorine at this position to form this carbon-nitrogen bond. You can then reduce that nitro group, which I believe you've been introduced to in previous seminars, through a multitude of different ways, such as hydrogenalysis, ion reduction, but here we found the most facile route was through zinc reduction. Additionally, we conducted this under a nitrogen atmosphere because the inclusion of oxygen led to oxidation across this bond and imine nucleophilic attack of that imine to form this byproduct. So to avoid that, we had to exclude oxygen from this reaction. So looking at the construction of this scaffold, quite simply, you can perform this from this commercially available aminotriazole molecule, where you simply heat this in the presence of this dicarbonyl compound here. And so it's a bis condensation reaction, and you can easily form this this dimethyltriazole in that process. And the final step is amide bond formation, again using an activated benzotriazole derivative in presence of base to form the carboxamide. So I thought I'd show you another little target-based screen that we did looking at blocking erythrocyte invasion. And here we're looking at a protein-based biochemical screen where we have an RH5 protein, which is on the surface of the myristoyte, and basogen, which it interacts with on the surface of the erythrocyte. So we can block or find binders that interfere with this interaction. We'll be able to block the myristoyte invasion of the parasite into the erythrocyte. And so the hit compound we got out of this screen against a known drug-like library was peronoclast, which is a leukotriene inhibitor, which is typically used for anti-inflammatory diseases. And you can see here it has a very modest EC50 value against RH5, and that's quite typical of a protein-protein interaction. And very weak activity against the parasite, but we're able to show that the activity that was observed against the parasite was in line with blocking erythrocyte invasion by the myrizoites, so consistent with the mechanism of action. So looking at the retrosynthetic analysis of this molecule, you can imagine several disconnection points here, an alkylation of this oxygen, carboxamide formation again. And quite typically, you'll look at this chromane molecule and make the disconnection point across this bond here with some type of protein-mediated coupling. But where, in matter of fact, you can construct this chrominane rather, from a condensation reaction, which I'll show in a moment's time. So quite simply, for this component of this molecule here, you take this phenol and then you perform a nucleophilic substitution onto this activated alkyl halide. So a simple displacement mechanism in presence of base, which gives you the phenolate. And then you want to go about to deprotect the carboxylic ester. So we didn't want to have the acid present here, because we also would have got alkylation of this carboxylic acid. So that's why we protected this with an ester. But now that we've formed the alkylation, we want to deprotect the ester, and we do that with base hydrolysis mechanism, which is simple nucleophilic attack of that carbonyl with this hydroxide anion. And then formation of this carboxamide, we want to activate that carboxylic acid in some manner. So we've refluxed that in thionyl chloride, formed the acid chloride quite simply, and that's our activated carbonyl. And then we react this with this aniline here, which is a precursor to this chrominone. And so that goes relatively straightforward, just by nucleophilic attack with that carbonyl and leaving group with that chlorine in the presence of base. And then we want to perform this condensation reaction with this ester group here attached to this tetrazole. And so the reaction mechanism here is we have this strong base. So this is sodium terbutoxide, which effectively forms an enolate here. So a carbon anion, which goes on to attack this carbonyl and the ethoxide as the leaving group to form this malonate here. So once we have this system, we're in place to form this acidic condensation reaction, which happens in these under refluxing conditions to get to our target molecule here. And that's quite easily isolatable. And so that's the synthesis of that example. So I'll move on to our final malaria example, which is these plasmepsin inhibitors. So just to give you a bit of background behind aspartyl proteases, of which plasmepsins are. So these aspartyl proteases contain these DTSG sites, which are these catalytic aspartyl dyads. We have two catalytic aspartates that perform the cleavage of any peptide substrate. And so these perform the peptide substrate between the P1, the P prime sites of the peptide of interest. And these peptide or amino acids in the peptide bind to the S pockets of the protease of interest. And so in Plasmodium, there's 10 of these cathepsin D-like aspartyl proteases known as plasmepsins, and these are highly conserved in all Plasmodium species. And these plasmepsins have a distant homology to human aspartyl proteases, so we should be able to design small molecules to target these plasmepsins over the human aspartyl proteases. And so in the asexual stage of the malaria parasite, we know that plasmepsin 5 is essential through genetic studies, but we also know that plasmepsins 9 and 10 are essential through genetic studies. So these are the plasmepsins that we want to be targeting in the malaria parasite. So in these genetic studies, we're able to delineate that plasmepsin 5 is required for protein export. So when the malaria parasite invades the host erythrocyte, it forms this parasitophorous vacuole around itself, which segregates itself from the red blood cell. So the parasite wants to remodel the host red blood cell for sustenance for its own development. And to do so, it must export proteins into the host red blood cell to manipulate the host red blood cell. And it does this via each of these proteins having an N-terminal label known as a plasmodium export element, or a PEXL motif for short. And so this PEXL motif is this amino acid sequence here on the N-terminus of these proteins, known as RXL, X, E, Q, or D. And plasmodin 5 is known to cleave between the L and the X of this sequence. And only then are these proteins then licensed for export into the host red blood cell. And so a small molecule inhibitor we hypothesized would block this PEXL processing event, block protein export, and bring about parasite death. And so what we did was design molecules that directly mimic this RXL motif, or the PEXL motif. An example is shown here. So cannabinine is an arginine isoster. We have valine, and we have this L-statine molecule. So this is a mimic of leucine. But additionally, we have this hydroxyl motif in this molecule, which binds to the catalytic aspartates, inhibits and function. You can see here that this molecule inhibits plasmodin 5 at a biochemical level very potently. And we're also able to obtain a crystal structure of this molecule bound to plasmodin 5, which informed further optimization design. We're able to show that this molecule inhibits PEXL processing. We're also able to show that it inhibited protein export and also killed the malaria parasite in vitro. Additionally, we're able to show that these compounds that target plasmodin 5 also impacted the transmission of the form of the parasite, the gametocyte, back to the malaria parasite. So in other words, we could design a transmission blocking agent here, also targeting plasmodin 5. So we'll be able to show these molecules blocked gametocyte-specific processing of proteins, blocked gametocyte death, but also blocked transmission of the parasite back to the mosquito, shown here in a laboratory setting. And finally, we're able to further optimize this molecule by concentrating on the P2 region of the molecule. So introducing a cyclohexyl ring here. So here you can see we're going from 842 to 601, improving potency in a biochemical context, but also with PEXL processing and against the malaria parasite. We're also able to obtain a crystal structure of this molecule bound to plasmodin 5, showing its mechanism of inhibition of this protein. So retrosynthetic analysis, so you may gather this is very peptidic-like molecule. So at the heart of some of Rani's lab's syntheses, this is a very small peptide by nature. So in this instance, we didn't go to solid phase solid phase peptide synthesis because it's such a small molecule. We thought we'd just do it by solution phase, but same principles apply with protecting group strategies. So as you can see, a lot of disconnections across amide bonds here, but this means that we require orthogonal protecting groups along the route in this synthesis. So here we're introducing this BOC-statine group analog, which can be ordered from a commercially available vendor. We form a amide coupling reaction, which I've introduced to you before, to form this BOC protected statine product. Then we deprotect the N-terminus of this building block to give the free amine, which is available for the next peptide coupling reaction, of which we couple onto another BOC protected carboxylic acid, and so forth. We go on to deprotect that molecule, and again we introduce the final cannabinine arginine isoster building block onto this molecule to give the final molecule. So a lot of repetitive steps in the synthesis, but that's fairly common with a peptidic-like molecule. So finally, I would just like to move on with malaria examples of targeting plasmodin 5, 9, and 10. And so these plasmepsins are very important for the invasion process and the egress process in various stages of the malaria parasite's life cycle, in terms of the blood stage, in terms of the transmission phase of the malaria parasite, that is the gametocyte, and also in the liver stage. And so these plasmepsins are key to processing very important invasion and egress ligands or proteins that are involved in these processes. So these cleavage processes don't take place by these plasmepsins. The parasite isn't either able to invade or egress from the malaria parasite. So in inhibiting these plasmepsins, we kill the malaria parasite. So we performed initially a screen against the malaria parasite using an aspartyl protease drug-like library. We identified this representative screening hits here. We then did mechanism of action studies and determined that they were targeting plasmepsins 10 quite potently in these instances. But we then went on to optimize these molecules, and we were able to create dual inhibitors of both plasmepsin 9 and 10. So very potent inhibitors. You can see here we're down to 60 picomolar in range against plasmepsin 10. We do have selectivity against human proteases, but also plasmepsin 5. But very potent compounds against the parasite. We hear sub-nanomolar potent levels. And so this was a very early lead compound, which we're calling a tool compound. And so we're able to show these compounds cleared parasites in the asexual stage. We're also able to show these compounds cleared parasites in the liver stage, that is, they didn't progress to the blood stage. And we're also able to show that in a laboratory setting that we're able to block transmission to the mosquitoes. So a multiple stage acting antimalarial. And these antimalarials, given they target the two plasmepsins, have a very high barrier to resistance, which is exactly what we want in an antimalarial drug. And so this work is moving towards the clinic. But today I'm just going to show you synthesis of this tool compound here. So you can envisage the disconnection between this carbon and nitrogen bond here and this carboxamide here. But also show you construction of this aminopyrimidone. So the construction of this aminopyrimidone is utilizing what is known as this sulfonamide chemistry, which is really chemistry that's come out of or originated from John Ellman's laboratory. And so in this instance here, we have two ethyl groups, which are obviously achiral because they're obviously the same. But typically this synthesis is used to install asymmetry at this site. So for example, if we had one of these substituents as a phenyl ring, we'll be able to do this in a stereoselective fashion using this synthesis. And this is what this synthesis was designed to do. So here we have this ketone and this sulfonamide rather, and quite simply this titanium oxide acts as a Lewis acid to activate the nucleophilic attack of that amine onto this ketone at a condensation reaction. Then we have this sulfonamine, and then what we want to do is create an enolate using LDA, which is a very strong base, to deprotonate this this hydrogen on the carbon just here, form the enolate, and then this attacks the amine to form this group here. And this titanium isoprop oxide is used to as a chelating reagent to direct the direction of the face rather of this enolate of the incoming nucleophile onto this imine if we had a large bulky group in this position, of which on this molecule we don't. And that's the reason why we get stereoselective outcome in this reaction usually. So now we have this molecule here. We then go about to deprotect this sulfonamide rather. sulfoxamine, rather. I should get my terminology correct, which is the beauty of this John Ellman chemistry. And then what we do is we go about to activate this guanidinium group here to nucleophilic attack by this amine. There's multiple steps in here, sorry about that. But the first step is this amine attacking this sulfonamine group, this activated sulfur group as a leaving group. And then we get concomitant cyclization in this reaction to this heterocycle. So the paramethoxybenzyl group may not be a group that's been previously introduced to you, but it's also a protecting group that can be simply moved by hydrogenolysis, which is the step in this reaction here. So that gives us this aminopyrimidine building block. So the next step is to install this central component, or at least construct it. And we do this from this commercially available ketone and perform a simple reduction here. And there's many reduction processes that we've used here, such as a Wolff-Kishner ketone reduction, which you've been introduced to in your curriculum. And then we perform what is known as a Mitsunobu reaction to affect this carbon-nitrogen bond formation here. And this simply couldn't be performed by an alkylation step, because we would always get alkylation on this nitrogen. And this was very confounding in the first instance when we first performed this chemistry. But Mitsunobu cleanly affects this carbon-nitrogen bond formation selectively. And then we perform chiral chromatography to elute this anandamide specifically, which is the active anandamide. And then we perform a hydrolysis of this methyl ester. So this is potassium trimethylcyanate, which is obviously an anion, which attacks this carbonyl bond here, which performs an intermediate trimethylsilyl ester. And in the workup, you add in some type of acid, and this hydrolyzes the TMS ester to give you the carboxylic acid. Then we perform an abide coupling using these conditions that we've introduced before to form this carboxamide bond, and obviously deprotection of this Bock group as we've introduced before with acid. And then, just looking at my time, Rani, have I got another few more minutes, or do you want me to stop here and let the students read on? Or
if you have several slides to go, it's okay. You can complete the slide first, and then we can continue the discussion later.
Okay, so maybe I'll just introduce schistosomiasis, and then the HIV example I'll just leave off the end here.
Okay.
And I think schistosomiasis is probably relevant to Indonesia as well. So I think it might be important to mention. And it's a disease caused by male and female schistosomes, as shown here. And it's endemic to 70 countries worldwide and affects approximately 2 million people. And a really an unfortunate death rate is reported annually also, which is totally unacceptable. And so the symptoms of getting this disease include fever, a rash, and abdominal pain, given where the parasite infects the human host. And without treatment, these symptoms can last for up to 8 years, which is the lifespan of these schistosomes. And ultimately, if left untreated, you can get liver and kidney failure, anemia, malnutrition, and can lead to death in some cases as well. And so schistosomiasis life cycle, very briefly, has this snail-like host, which is aquatic in nature, and this is where the disease effectively stems from. They produce these free-living or free-swimming crochyria, which then go on to penetrate the human host through the skin. So typically in rice fields or in aquatic areas where human hosts might go into play, this is where you get infection of this or transmission of this schistosome. And so these schistosomes invade initially into the liver, but then make their way into the intestinal tract of the human host. And this is where they cause a lot of different issues, as mentioned on the previous slide. And eventually the eggs of these worms are secreted and the life cycle continues. And so there's only really one current treatment approved, which is praziquantel, or PZQ for short, but resistance has emerged against this drug. And there's really no new treatments emerging, perhaps apart from moxidectin, which is used to treat worms in dogs. And so the advent of genome sequencing has really provided, of the schistosome worm, has really provided new druggable protein targets. And in brief, one of the druggable targets that we're working on is the BCL2 family, which is known for driving apoptosis in humans. And so we had a BCL-XL program at our institute looking at targeting cancer via these BCL family proteins. So we're able to screen these small molecules that we had in this BCL-XL human program against the schistosome host. And we discovered this molecule here, which had sub-micromolar activity against the schistosome BCL2 protein in a biochemical assay. We're able to use structural studies to then inform further molecular design, and found that this part of the molecule was key to driving optimization. As you can see, we're down to 30 nanomolar against the schistosome BCL2 protein. We're able to then show that we could affect apoptosis in artificial cells that were dependent on schistosome BCL2 and kill worms at very modest EC50 values as well. And this work is at a very early stage and ongoing at the moment. But just finally, to look at this key last example of synthesis, looking at the retrosynthetic analysis, you can envisage amide coupling here, a condensation across this carbon nitrogen double bond here. Between this carbon-carbon bond here, perhaps a Suzuki reaction, and this carbon-oxygen bond here, an alkylation. So starting off with this 2-chlorothiazole, that has a carboxylic acid in the 3 position, we can envisage an amide bond formation here, which we had issues isolating because of the hydrolysis of this chloro group, so hence the low yield. But we could then perform a nucleophilic substitution with hydrazine at elevated temperatures. So again, just a simple nucleophilic aromatic substitution of, and that chlorine acts as the leaving group. Very briefly, we can then build up this part of the molecule by first protecting this carboxylic acid with an ester group, which we needed to do because we didn't want to have this functionality going onto this carboxylic acid. We then performed a bromination reaction, which goes at this position selectively because this OH group is the directing group towards this position here. And so that's obviously a type of reaction you've been introduced before. And then we perform this alkylation reaction, which we're using in this instance a Mitsunobu reaction again, but you can use an alkyl halide quite simply here to affect alkylation of that oxygen atom. And then we need to form this carbon-carbon bond here. And so to do this, we perform the Suzuki reaction, which uses an aryl halide, but also, an aryl halide here, and also a boronic acid. But first we need to perform that boronate in this instance. And so we use palladium-mediated coupling reaction to form that boronate, and then we go on to use this Suzuki reaction to form this carbon-carbon bond. I believe Trent Ashton may have introduced this chemistry to you in a previous seminar. And so the last step is just a simple condensation reaction with this amine nitrogen onto this ketone here to form this hydrazone to get to the final molecule. And I think I'll leave it there in lieu of time. Sorry, I just went over a bit over time there. But there is one more example here on our work with HIV latency reversing agents, of which we have developed very potent molecules and been able to show that they work in donor cells from HIV infected patients, and the retrosynthetic analysis, which is shown here, which is fairly simple for you to follow. And finally, thank you for your time and attention, and happy to answer any questions. And thank you, Rani, for inviting me and hosting me.
Yeah, thank you so much, Brett, for very interesting lecture. We can learn how the drug can be obtained through the drug discovery stages, and also you have give us many synthetic pathways for synthesizing some hit compounds for some infectious diseases. So I believe there's some question from student and other participant, and probably the question can be delivered directly to Dr. Bradley by using raising hand, and then you can ask directly to Brad, and also you can write your question in chat box. So yeah, I'm looking, yeah, I'm inviting for the question from the audience. Yeah, there are several questions here. The first question coming from Trigarin. Garin is the student, undergraduate student in UNPAD. The question is, what happens if we modify a drug using microencapsulation? Does it can increase the efficacy? Because with microencapsulation, we can control the absorption process. And based on your experience, which is the more specific and better drug delivery between the phenotypic approach and the target protein?
Yes, thanks for the question. And obviously, I didn't have enough time to go into delivery processes or formulation, but certainly microencapsulation is really important in drug discovery, particularly for drugs that aren't very well absorbed, as you point out, and need an additional formulation to assist with their absorption. So microencapsulation is a nice way to control the release of the drug as well. And there's many ways you can do microencapsulation, as I pointed out. So certainly it can increase the drug's efficacy if it increases the drug's absorption. And based on my experience with respect to which is better, a phenotypic approach or a target protein-based approach. So as I alluded to in the seminar, there's obviously advantages to either approach. So looking at a malaria perspective, which is, I suppose, a key focus of my laboratory, I'd have to say the phenotypic approach is far more accessible than a protein target-based approach. Just the rate of which you can get to a drug candidate through this approach is far quicker than the target-based approach. But in other processes that I've been involved with, or other diseases rather, the target protein-based approach is almost always preferred. And the reason is because you know exactly which protein you're targeting, how you're targeting it, how it binds to the protein, which helps with not only the medicinal chemist to design the drug, but you also know what the outcomes might be with hitting other proteins that are similar to the one that you're targeting. And moreover, pharmaceutical companies typically always want to have a target protein-based program rather than a phenotypic-based program, purely based on selectivity and toxicity that you may encounter. So there's no right or wrong answer to that question, but certainly in malaria research, by far, the phenotypic approach has been more successful.
So I hope it answer ya, Trigarin. Yeah, another question is coming from Andika Rizki. He is also a student at Unpad. She wants to ask about de novo, which is one of the starting point in the identification method. Can you please explain more about this method? And is this method been used before? If it is used, how is the mechanism?
Yeah, so the key example that I gave for that approach, and I've sort of coined it de novo because you're starting from the natural substrate. So the key example I gave was the plasmepsin inhibitor. So where we knew what the amino acid sequence was that plasmepsin 5 cleaved, which was that RXL motif, and we modeled our molecule based on that natural substrate. And so I suppose de novo is not the terminology that's typically used, but perhaps substrate mimetic would be the terminology that's more typically used there and you might be more familiar with. And so that's the key example that I was giving with that approach. So again, not all drug discovery programs start with screening methods of small molecule libraries. You can also develop small molecule inhibitors from the natural substrate, which is, also a common method of how drugs are designed.
Yeah, I hope it answer Andika. So I have another question from Reza. Reza is also a student from Unpad. She wants to ask about drug development process, specifically in clinical tests for the drugs. Have you ever experienced some drug that work well when used in the animal, but it was fail when tested on human? If yes, why it can happen and how to overcome it?
Yeah, so I suppose at that point, we would have already been collaborating with a pharmaceutical company. So once it gets to the late lead stage and moving into humans, basically it's out of the early phase drug discoverer's hand, which is the area that I'm in, and it moves into the pharmaceutical area. So my experience here is, I would say, indirect. But what I can say is that there's multiple reasons why the drug may work in an animal model, but not in humans. And one reason is there's different processes that are involved in a mouse versus that in the human. So there's different homology between the protein target that you may be targeting in both animal versus human. You may have different metabolism processes in a human versus a mouse, so how the drug is turned over. And also different absorption and elimination mechanisms between the two species. And so this is a reason why the drug might not be as systemically exposed or efficacious as one would have predicted it would be from what data you have in an animal model versus what data would be seen in a human. Additionally, there's another factor, which is adverse events in the human, which is generally the reason why drugs fall out of clinical trials. So obviously going into humans, your safety margins have to be quite large, because any small adverse effect in a human clinical trial will automatically end that human clinical trial almost immediately. And obviously there's reasons why you would do that for obviously safety reasons for that human subject that the trial's been performed on. And so typically it's these unforeseen adverse effects or adverse events that happen in human clinical trials, the main reason for drugs falling out of clinical trials, which aren't seen or observed in animal models as well. So I suppose in summary of that, two reasons: the molecule not being efficacious enough in humans as it was predicted to be in mice, and the second reason is for toxicity reasons, which are unforeseen and didn't occur in mice. and hence why there's a large attrition rate in human clinical trials with drug, and why drugs cost a lot of money to develop, generally $1 billion US, and if not more. So you can see why pharmaceutical companies want to get some money back when their drug gets to market, when it costs that much to develop a drug.
Yeah.
Yeah.
I think it has answered your question, Reza. So okay, thanks for your answer. So I'm still inviting question. You can participant can ask directly or also can write the question through chat box. I think there's another question from Aldrin and Lenga. So I will read a question from Aldrin first. Aldrin is also a student from Unpad. She wants to ask about the drug discovery process. How should the treatment be when a candidate of drug are tested on human and it turns out to be effective enough for some people but gives side adverse effect for some others? Is it possible for some people to resistant to some drugs during their period using those drugs?
Sorry, I got interrupted, Rani.
Okay.
Go on.
Okay. And how to overcome that?
Sure. So most definitely, side effects and the efficacy of drugs in certain individuals can be completely different to other individuals in humans. This is, and even in mice, actually, in certain different cohort species. And this is the reason why once you get to phase 2, phase 3 clinical trials, you want to test your drug against as great a cohort of people with the greater diversity of background as possible. Because in some human subjects, the drug might be metabolized differently to others. And you'd be surprised at how different metabolism is between different humans. And then obviously you can see the difference with some humans with respect to obesity and versus other people that seem to be able to eat anything they want and still not put on weight. So that just gives you an idea of the different metabolism that different humans have, and that still applies to drug discovery too. So this is, again, the reason why it's very important to test the compounds in phase 3 clinical trials before going to market against as many people as possible with a wide range of genetic backgrounds for that very reason. It's obviously predicting it before you get to human clinical trials is the key, as I pointed out before, with efficacy and adverse effects. And I suppose at a very basic level, some of the models that I gave as examples in the slides should be applied at a very early stage in drug discovery to give you an idea of what or how your drug is going to be turned over and absorbed in the human host, but also to try and predict what adverse effects your drug may have moving into the human host. So it's always important to apply these principles very early on in the drug discovery process, so you don't get into the position where the drug may work in some humans over other humans. But of course, you want to obviously selectively target the disease in some individuals, but not affect others. So it's all part of the drug discovery process to try and predict as best as possible.
Yeah. Thanks, Brad. And then I hope it has answered your question, Aldrin. And the next question is coming from Lenga. Lenga is our alumni from our department. Mm-hmm. She wants to ask regarding the interaction between the active compound, whether the interaction is predictable, whether how to predict its to predict it's the same as the interaction of molecule in a compound, because in a drug is made of several active compounds, and to maintain the stability, it's very difficult. Can you get the question?
Yeah, I can. Yeah, so very much so. It's very difficult to predict the metabolic stability of a compound. And again, in the additional slides that I gave, there are in vitro tests that we can do where we incubate the compound in liver microsomes to determine how stable the compound is before going into animals. Additionally, we can also incubate the compound in human plasma as well to tell us about the stability. We also do chemical stability tests as well to make sure the compound is stable in gut acid and gastrointestinal circumstances too. We generally do this all before we go into the mouse model. And the reason is because we don't want to be testing every candidate we make in a mouse model for ethical reasons primarily. But certainly there are processes and assays we undertake to assist in us understanding how the compound might be, again, absorbed in an in vivo system, metabolized, and also excreted.
Yeah. I hope it has answered your question, Yalenga. We still have another question, Brad. It's coming from Harum. Harum is also a student from our university. She wants to ask about some challenges when you are discovering potent drugs, especially anti-malaria drugs, and how did you overcome some of the obstacles?
Yeah, so on metabolism, some of the examples that I showed, we couldn't develop further because of a key metabolic event across a functional group that was utterly required for malaria activity. And so many of the hit compounds that we discovered, we can't advance all of them because they either have inappropriate physiochemical properties that lead to them having poor absorption, or they're metabolically turned over. And so, again, this is effectively like the attrition rate in the clinic. At the early drug discovery phase, there's also an attrition rate where certain aspects of the small molecule can't be overcome. So we effectively stop working on that molecule to prioritize our resources on a more promising molecule. And so we try to overcome these liabilities as best as possible using the medicinal chemistry techniques that we know. And if you read some of the articles that I gave reference to today, you can sort of get an idea of some of those. But again, in some instances, it's just not possible to overcome this, and we do stop working on that molecular scaffold. This not only applies to animal research, but also other drug discovery programs too.
Yeah, thanks, Brad. Another question from Rizka. Rizka would like to ask a question about how lipophilicity can be affected and considered in drug discovery. Thank you very much.
Yeah, so lipophilicity, as I said, is conversely related to polarity. So you don't want your drug to be too lipophilic, but you don't want your drug to be too polar. So as I mentioned, polarity will assist in your molecule being water soluble. But if your molecule's too polar, then you won't have membrane permeability for it to get into the cell or be readily absorbed intestinally in an in vivo system if you dose it orally. So that's the reason why the balance between lipophilicity and polarity is just so important to get right in a drug discovery program. So again, if you I showed that plot with the absorption, which relates to cell membrane permeability. If you have a look at that, you can see the the area of lipophilicity that you want to be in with respect to your small molecule for your molecule to be readily absorbed. But outside of that, you're not going to get good cell membrane permeability, or it won't be readily absorbed. So it's, again, it's the balance between lipophilicity and polarity in your molecule, as mentioned in the seminar. And it's a real balancing act, and one that the medicinal chemist has to fight with. It's almost a constant battle, actually, with respect to those parameters.
Thanks, Brad. I think we have our last question. It's coming from Dwi Nur Indah. She's curious about malaria drug. Why so many drugs with different structure can cause the same activity? Probably, yeah. Why
Yes,
it is so? Yeah, thank you. And
yes.
I think the same. Yeah.
Yeah. So, as is the case in humans, we have we're made up of a lot of different proteins, and those proteins are important for the development of a lot of different facets of our biology. And so the same rule applies in malaria parasite. The parasite has a whole host of different proteins, and a lot of those proteins in the malaria parasite are essential for its development and human pathogenesis. And so inhibiting or interrupting the mechanism by which those proteins operate is the reason why we can have a lot of different chemotypes targeting the malaria parasite. And so obviously those compounds bind to give these different proteins in different ways, because the surface of the protein between proteins is so significantly different. So the old keyhole type mechanism applies here, where you have different proteins require different small molecules to bind to the correct pocket in the protein and to inhibit their action. And that's the reason why we can have so many different chemotypes that target essential processes in the malaria parasite, as we can have small molecules that target different protein targets involved in disease pathogenesis in humans.
Yeah, I think that's answered all of the question, but since we have limited time and there's, I think, no more question from the participants. So thank you, Brad, for the lecture today. Yeah, we have learned many things, not only how the synthesis, but also how the drug can be obtained. And of course, we can see how where is our position as like organic synthesis chemist in this drug discovery process. So before I close, is it okay that we can like take a picture with the participant?
Yeah.
I think I will ask all of the participants to open the camera. So we will take a picture together with Dr. Brad Slips. So yeah. Yes. Yes. Yeah. I think there's like almost 81 participants has joined this lecture. So I'm waiting for all of the participants to open the camera. Okay.
Yeah. Like you got a screen.
Yeah. Yeah. Okay. I think I'm going to start with the first slide. 1, 2, 3.